Now that we have established who created this virus, we enter the shadowy realm of viral genetics. One concept emerges as critical to understanding the complexity and sophistication of SARS-CoV-2. And that is open reading frames, or ORFs. These ORFs are segments of genetic code that serve as the blueprints for proteins which are essential for the virus's structure and function. They dictate how the virus replicates, how it evades the immune system, and how it spreads from one host to another. For SARS-CoV-2, these ORFs are not just markers. They are the signature of the virus's creators and the architects of its existence. Understanding these open reading frames, ORFs, could provide the key to decoding the virus's behavior, paving the way for targeted treatments. By mapping these genetic blueprints, researchers can predict mutations and develop more effective strategies to combat the virus. The study of ORFs is not merely an academic exercise, it is a critical step in uncovering how this bioweapon has affected us all and identifying potential ways to mitigate its impact. Open reading frames, or ORFs, are crucial elements in the genetic code of organisms, including viruses like SARS-CoV-2. These sequences are fundamental to the way viruses operate, dictating everything from their replication to their ability to evade the immune system. Understanding ORFs is akin to deciphering the very blueprint of viral existence. An ORF is a sequence of DNA that has the potential to be translated into a protein. Proteins are the workhorses of the cell, performing a vast array of functions necessary for the virus's survival and propagation. Without these proteins, the virus would be unable to replicate or infect host cells. It starts with a start codon and ends with a stop codon without any interruptions by stop codons in between. This uninterrupted sequence is essential because it ensures that the entire protein can be synthesized correctly. Any interruptions could result in a non-functional protein, which would be detrimental to the virus. This uninterrupted sequence allows for the synthesis of proteins which are essential for the virus's structure and function. Proteins form the building blocks of the virus, making up its outer shell, its enzymes, and other critical components. They also play a role in the virus's ability to hijack the host cell's machinery for its own replication. In simpler terms, think of an ORF as a recipe in a cookbook. Just as a recipe provides the instructions for creating a dish, an ORF provides the instructions for creating a protein. Each step in the recipe corresponds to a specific part of the protein synthesis process. The start codon is like the title of the recipe, signaling the beginning of the instructions. It tells the cellular machinery where to start reading the genetic code. Without this start signal, the process of protein synthesis would not begin. The stop codon is like the end of the recipe, indicating that the dish is complete. It tells the cellular machinery to stop reading the genetic code, ensuring that the protein is synthesized to the correct length. If the machinery were to continue reading beyond this point, it could result in a malformed protein. Everything in between are the steps needed to create the final dish, or in this case, the protein. Each step in the ORF corresponds to a specific amino acid in the protein, and the sequence of these steps determines the protein's final structure and function. For SARS-CoV-2, these ORFs are not just markers. They dictate how the virus replicates, how it interacts with host cells, and how it evades the immune system. Each ORF has a specific role in the virus's life cycle, making them critical to its survival and pathogenicity. Understanding ORFs is like understanding the blueprints of a building. Just as a blueprint provides a detailed plan for constructing a building, an ORF provides a detailed plan for constructing a protein. Each part of the blueprint corresponds to a specific part of the building, just as each part of the ORF corresponds to a specific part of the protein. Each ORF has a specific role, much like different parts of a blueprint contribute to the construction of a building. Some ORFs are responsible for creating the virus's outer shell, while others are responsible for creating the enzymes that help the virus replicate. Each ORF is essential to the virus's overall structure and function. By studying these ORFs, it is hoped scientists can gain insights into how the virus operates and how to combat it. For instance, it is hoped that in understanding how ORF3A inhibits autophagy can help researchers develop strategies to counteract this effect, potentially leading to new treatments. For instance, ORF1A and ORF1B in SARS-CoV-2 encode for proteins that are involved in the replication of the virus. These proteins are essential for the virus to copy its genetic material and produce new virus particles. Without these proteins, the virus would be unable to replicate and spread. ORF3A, on the other hand, is known to inhibit autophagy, a process where cells digest their own components, thus helping the virus persist within the host. By inhibiting autophagy, ORF3A allows the virus to avoid being destroyed by the host cell's defense mechanisms, ensuring its survival and continued replication. ORF6 suppresses interferon responses, which are part of the immune system's defense mechanism. Interferons are proteins that help coordinate the immune response to viral infections, and by suppressing these responses, ORF6 helps the virus evade detection and destruction by the immune system. ORF8 modulates apoptosis or programmed cell death to ensure the virus's survival. Apoptosis is a process where cells self-destruct in response to damage or infection, and by modulating this process, ORF8 helps the virus avoid being destroyed by the host cell's defense mechanisms. ORF11, developed by China's WIV, enhances viral entry into human cells. This ORF encodes a protein that helps the virus attach to and enter host cells, making it easier for the virus to establish an infection. By understanding these roles, scientists can hopefully develop targeted therapies and treatments to combat the virus. For instance, treatments that stimulate the immune system to recognize and attack the proteins encoded by ORF6 could one day help prevent the virus from evading the immune system. In summary, open reading frames are like the blueprints of a virus, dictating its structure, function, and interactions with the host. They are essential for the virus's survival and pathogenicity, making them a key focus of scientific research in the fight against SARS-CoV-2. Together, these ORFs form a sophisticated and deadly toolkit that allows SARS-CoV-2 to navigate its host environment with unparalleled efficiency. Each protein encoded by these ORFs serves a specific purpose. Some inhibit the immune system's response, others enhance viral entry and replication speed, while some modulate how cells respond to infection. The convergence of these genetic elements into SARS-CoV-2 exemplifies an unprecedented level of biotechnological sophistication. It is a virus that embodies the best and worst of human ingenuity, born from a collaboration spanning continents and decades. As the world grapples with the legacy of SARS-CoV-2, the need for transparency and accountability has never been more urgent. Our research has exposed gaping vulnerabilities in our global health systems and highlighted the importance of open communication and cooperation among nations. It becomes clear that the origins of this pandemic are deeply intertwined with the shadowy world of bioweapons research. This revelation must spark a global outcry for more stringent regulations and oversight in scientific research, particularly in areas that have the potential to impact public health on such a massive scale. The discovery of all of these ORFs combined in one virus underscores the need for greater transparency and accountability in international scientific collaboration. Scientists and researchers must work together, sharing data and findings openly to ensure that the benefits of scientific advancements are realized without being weaponized, compromising safety and ethical standards. Lest humanity again finds itself ensnared by the unintended consequences of its own ingenuity, it is imperative that we learn from this pandemic. By fostering a culture of transparency and accountability, we can better prepare for future challenges and safeguard the health and well-being of us all. For more on the origins and implications of SARS-CoV-2, including research performed in top-secret projects carried out in our growing global network of labs, continue watching as we unravel the complex threads of this viral saga.